Hi everyone. So in this week's webinar, we're going to be looking at the cell membrane and specifically different types of cell transport mechanisms. So firstly, the membrane structure is really important for controlling the exchange of materials between the internal, so inside the cell, and the external, which is outside the cell, and those environments. Now, the membrane structure consists of what we call a bilayer. Now, bilayer means two because we have two layers in here that make up this cell membrane structure. Now, this little structure here is called a phospholipid, and there are two lots of phospholipid layers, which is why it's called a bilayer. Now, they have a hydrophilic end, which means hydrophilic means water loving, and a hydrophobic tail, which means hydrophobic means water hating. So the water hating parts, the tails, orientate inside the cell membrane structure, and the hydrophilic heads are on the outside. Now, we also have these things called membrane proteins that span the width of the membrane, or they can sit in the bilayer. So here we have a membrane protein that spans the entire width of the membrane, whereas this one only is partially embedded in the membrane structure. Now we also have glycoproteins as well, and they are carbohydrate change. Now we don't actually have any in this diagram here, and we've also got glycolipids as well, and these basically help with the structure of the membrane. Okay, so all of the materials that pass through a cell membrane can be controlled to move into or out of a cell. And these include gases, nutrients, and waste products. Now, these factors we have three main factors that control the movement of materials into and out of a cell, and they include size, charge, and solubility. So size means that only really small molecules can be exchanged freely between the internal and external environment of a cell. If you want to move larger molecules, they need to have help, and usually that help comes in the form of a protein channel because they can't move freely. In terms of charge, some atoms or molecules will have a charge, and that charge allows them to be exchanged from one side of the membrane to the other. And usually, again, this is through transport proteins like this one here that allow movement of charged molecules or atoms from one side of the membrane to the other. Now, the last one is solubility in water. Now, only molecules that are not soluble in water will be able to pass through the membrane because we have these hydrophilic tails here that don't like water, so they won't allow molecules of water to pass through that membrane. So they have to move through channel proteins as well. Okay, so moving on to different types of movement of materials in a cell, the first type of movement that we're going to look at is diffusion. So diffusion is classified as what we call a passive transport process, and that means that it doesn't require any energy to occur. Now, diffusion moves along what's called a concentration gradient. So you will have two areas of concentration, an area of high concentration, which is here on the left, and an area of low concentration, which is over here on the right. In diffusion, these molecules, whatever they are, will move from an area of high concentration to an area of low concentration. Now, diffusion usually occurs along a partially permeable membrane. So what partially permeable membrane means is that only some substances are allowed to move across that membrane, so it can be selective in what it lets out of or into the cell.
Have you ever wondered what it might be like to be inside a cell? Imagine the genetic material, the cytoplasm, the ribosomes. You'll find those in almost all cells, prokaryotes and eukaryotes. Eukaryote cells, in addition, have membrane bound organelles. All of those structures and organelles have different functions, but cells are not isolated little worlds. They do have a lot going on inside them, but they also need to interact with their environment. It makes sense that to keep a stable environment inside themselves, otherwise known as keeping homeostasis, they must have some control on what goes in and what goes out of them. A very important structure for this that all cells contain is the cell membrane. By controlling what goes in and out, the cell membrane helps regulate homeostasis. It's the homeostasis king. Let's take a look at the cell membrane. You could have a whole course on the cell membrane itself. It has amazing structure. It has signaling abilities. But to stick to the very basics, it's made of a phospholipid bilayer. Bilayer means two layers, so you have these two layers of lipids. Now, these lipids, they're called phospholipids, well, they have a head that is polar, and they have a tail that is nonpolar, making them quite unique. Some molecules, they have no problem going through the cell membrane, and they directly go through the phospholipid bilayer. Very small, nonpolar molecules, they fit in this category. They're a great example. Like some gases, oxygen and carbon dioxide gas, those are great examples. This is known as simple diffusion. Also, it doesn't take any energy to force those molecules in or out, so it's known as passive transport. Simple diffusion moves with the flow, meaning it moves with the concentration gradient. Molecules move from high concentration to low concentration. So when you hear someone saying that something's going with the concentration gradient, that's what they mean. They mean it's going from a high concentration of molecules to a low concentration of molecules. Now, remember how we said the cell membrane is actually a pretty complex structure? Well, one thing we haven't mentioned yet are proteins in the membrane, and some of them are transport proteins. Some transport proteins act as channels. Some of these proteins actually change their shape to get things across. Some of them open and closed based on some kind of stimulus. All of these are good things because it's helping with molecules that may be too big to cross the membrane on their own, or molecules that are polar and therefore need the help of a transport protein. This is known as facilitated diffusion. It's still diffusion, and it still moves with the concentration gradient of high to low. It doesn't require energy, though, so it's also a type of passive transport. It's just the proteins are facilitating or helping things pass. Charged ions often require a protein channel in order to pass through. Glucose needs the help of a transport protein to pass through. In osmosis, for water to travel at a fast rate across the membrane, it passes through protein channels called aquaporins. So these are all examples of facilitated diffusion, which is a type of passive transport and moves with the concentration gradient of high to low concentration. Now all the transport we've mentioned has been passive in nature.
So osmosis is another type of passive transport, and it is where one type of molecule moves either inside the cell to outside or vice versa. So this molecule is water. So water moves across the partially permeable membrane, again from a region of higher concentration of water to low concentration of water. But when we talk about concentration gradients, we actually talk about concentration of solute. So I'm going to show you a video to explain what solute is in much better detail than I could. and hopefully you'll understand.
You know what word gets thrown around a lot, but is actually pretty challenging to understand? Osmosis. But it does answer some great questions like why would it be deadly to give someone an IV of pure water? Or why would a saltwater fish placed in fresh water experience major problems? We'll answer these questions as well as explain the process of osmosis. Now, first, understand that osmosis is the movement of water, and that really is a type of diffusion. Often, when you're talking about osmosis, you're talking about the movement of water through a semi permeable membrane like a cell membrane. A cell membrane has openings small enough for water molecules to pass, but it will not allow larger things like salt molecules to freely pass, at least not without assistance. Osmosis is a type of passive transport, meaning that it doesn't require energy. I mentioned osmosis is a type of diffusion, and similar to what you might have learned about with diffusion, molecules, in this case water, travel from areas of high concentration of water to low concentration of water. But there's another way to think about water movement in osmosis. A low water concentration likely means there are a lot of solutes. Solutes are substances like salt or sugar that can be dissolved within a solvent, like water. Water has the tendency to move to areas where there is a high solute concentration, which means there's less water concentration. So if you want to easily figure out where the water will travel, look to the side where there is a greater solute concentration. So let's imagine you have a box with a semi-permeable membrane in the center. And remember, this membrane will let tiny things pass through it like water and gases, but not large solutes like salt. We'll label one side of the box A and one side of the box B. If you had pure water on both sides of the box, those water molecules, they're moving. Water molecules, they like to move. But the net movement of water on the two sides is zero, meaning the overall change in direction of movement is zero. Now let's imagine on side B you dump a huge amount of salt there. Salt molecules are too large to pass through this membrane. So which direction will the water move towards? A or B? The answer is B. This area has a high solute concentration, and remember, water moves to areas of higher solute concentration. You can almost think of the water as trying to equalize the concentrations, diluting side B. Once equilibrium is reached, the net movement of water on the two sides will be zero, but remember that water still likes to move, and movement still occurs. Here's some vocabulary to know.
So there are three factors that will affect both diffusion and osmosis, and these include the concentration gradient. So the greater the difference there is between the two concentrations, the greater the movement of travel. If the surface area to volume ratio will also have an effect on diffusion. If you have a greater surface area to volume ratio, then diffusion will occur at a faster rate. And the last one is temperature. If you have a higher temperature in a system, there's more kinetic energy in the system, and that means that the particles will move faster and bounce off each other more, and therefore this will decrease the overall movement of that particle substance covered by the particles. So we've talked about a mode of transport that doesn't require any energy, and that's passive transport, whereas active transport, as the name suggests, requires energy expenditure. And the reason for this is because materials are being moved against their concentration gradient. So we're moving materials from a low concentration to a high concentration of that type of molecule. So usually this requires carrier proteins that move those particles across the membrane against the concentration gradient. And we need some form of energy to allow this to happen, and that energy is ATP. So here we've got an example of a proton pump that is moving hydrogen ions from a low concentration to a high, from a high concentration to a low concentration. And in order to do that, you need ATP to be able to pump or fuel the pump to then move that hydrogen ion from the low concentration to the high concentration, which is against the concentration gradient. Now, there are two types of active transport that involve the membrane. So the first type is endocytosis. Now, endo means endo into, so this is movement of materials into the cell. So firstly, the materials are brought to the edge of the cell and usually up against the cell membrane, and a vesicle forms around those substances, and then that vesicle actually buds off from the cell membrane and carries the molecules into the cell, and then they're unpackaged in the cell and distributed to where they need to go. The other type of movement that involves the cell membrane is exocytosis. Exo meaning exit, so this is the movement of materials from inside the cell to outside the cell. So firstly, we have a vesicle that carries molecules. These could be waste products, it could be other materials that are needed elsewhere in the body, and it carries it to the perimeter of the cell. The vesicle membrane then bonds with the cell membrane, and this allows an opening to occur on the outside of the cell that then releases the contents of the vesicle into the extracellular environment to either be excreted as waste or transported somewhere else. Now, the last thing that we're going to talk about is the importance of surface area to volume ratio, and I think this video really quite nicely sums up the importance of that when we talk about transport of materials into and out of a cell.
The elephant is a creature of epic proportions, and yet it owes its enormity to more than 1,000 trillion microscopic cells. And on the epically small end of things, there are likely millions of unicellular species. Yet there are very few we can see with the naked eye. Why is that? Why don't we get unicellular elephants, or blue whales, or brown bears? To find out, we have to peer into a cell's guts. This is where most of the cell's functions occur, enclosed by a cellular membrane that acts as the doorway into and out of the cell. Any resources the cell needs to consume, or waste products it needs to expel, first have to pass through this membrane. But there's a biological quirk in this setup. A cell's surface and volume increase at different rates. Cells come in many shapes, but imagining them as cubes will make the math easy to calculate. A cube has 6 faces. These represent the cell membrane and make up its surface area. A cube measuring 1 micrometer on each side, that's 1/1,000,000th of a meter, would have a total surface area of 6 square micrometers. And its volume would be 1 cubic micrometer. This would give us 6 units of surface area for every single unit of volume, a 6-to-1 ratio. But things change dramatically if we make the cube 10 times bigger, measuring 10 micrometers on each side. This cell would have a surface area of 600 square micrometers and a volume of 1,000 cubic micrometers, a ratio of only 0.6-to-1. That's less than 1 unit of surface area to service each unit of volume. As the cube grows, its volume increases much faster than its surface area. The interior would overtake the membrane, leaving too little surface area for things to quickly move in and out of the cell. A huge cell would back up with waste and eventually die and disintegrate. There's another plus to having multitudes of smaller cells, too. It's hardly a tragedy if one gets punctured, infected, or destroyed. Now, there are